and mouse. Yeah, thank you.
Thanks. Actually, I also got my bachelor degree and also PhD in physics. And then in my postdoc, I transitioned to study single-cell genomics at Harvard Medical School with Alon Klein, where I developed CoSPAR to predict cell differentiation from single-cell lineage tracing data. And then since 2023, I started my own lab at Westlake University. So my lab currently has 9 people, including myself. 5 of them are working on computational side, and 4 of them are working on generating experimental data. So we are particularly interested in lineage tracing techniques and also their applications. So lineage tracing allows us to study the cell division histories within an organism. For example, this is the famous study in C. elegans using microscope. With lineage tracing, we can study, for example, how cells differentiate and how cells divide and proliferate, and also how cells migrate from one physical place to another. However, there are currently a great limitation in existing lineage tracing studies in humans. First of all, basically, I have to rely on somatic mutations, and one way to do that is to use whole genome deep DNA sequencing. However, this method because the somatic mutation rate is really, really low, it's 10 to minus 9 per nucleotide per division. So on average, only got 3 mutations per division. And also, it has very low throughput, can generate about 100 cells, with each cell cost about $200 US. And also give you only the DNA mutation information, typically do not have the allophenotypic information. A more recent way is to use the mitochondrial DNA mutations. However, there have been a lot of controversy, including the first 2 authors from the original 2019 Cell paper. They also raised a serious objection to a more recent Nature paper on the same topic. So we wanted to try something else, and then we got our inspiration from our previous work while we are working on these starting mice. So this is the collaboration with Dr. Lily at Westlake University and her former postdoc mentor, Fernando Camargo. And there we developed a starting mice that can have extremely high barcoding diversity and also very, very high capture efficiency and editing efficiency as well. At the same time, we also developed a single-cell chameleon-seq where for each cell we can get its gene expression, cloning accessibility, DNA station, as well as lineage barcodes. So I believe this is the first single-cell lineage tracing, multi-omic lineage tracing method. And applying this tool, we studied the blood development. So here we barcode the cells, the mice at the embryonic stage E10. And then the HCs will migrate to fetal liver and then go to the bone marrow. And then at this stage, about after 10 weeks, we collect all the HCs. And for each HC, we profile them using the chameleon-seq. So I'm very, very surprised by the data, because if you look at the gene expression, by the way, each color is an individual, a different barcode, different clone. So if you look at the low dimensional reduction on the gene expression data, you don't see really any pattern, and also from the cloning accessibility. But if you look at the DNA station, you can clearly see that cells are clustered by clonal identity. We call it clonal memory, and based on this data, we conclude that the long-term clonal memory is maintained only in DNA station, but not in gene expression or cloning accessibility. So this gave us a lot of inspiration. Maybe we can use DNA station to do lineage tracing. So after thinking more about DNA station, actually this makes a lot of sense, because in DNA station you have this so-called epi mutation. Basically, a DNA station happens on the CpG sites, and when cells divide, the DNA station profile can be replicated to their daughter cells. However, due to this replication, occasionally you make errors, and the error is about 10 to minus 4 or 10 to minus 3 per CpG sites per division. Remembering that in human genome, we have about 30 million CPG sites. So this gave us about 30,000 CPG mutations per division. And also, since the mutation is really, really low, so if we invert this, we give, it means that for each epi mutation, it could be stable for about 1,000 cell divisions. Basically, we rarely get 1,000 divisions in a human lifespan, so I guess this tool should be sufficiently stable to trace the lineages in the human body. So, however, there are substantial challenges to do lineage tracing based on single-cell DNA methylation data. First of all, if we use single-cell bispecific sequencing, there are about, it only captures 5% of the entire genome, or 95% of data is missing. Secondly, we know that when cells differentiate, different cell types, they develop cell type-specific DNA methylation pattern. So how to remove these cell type-specific methylation profiles is also a challenge. And lastly, we know that in the mammal development, so the global methylation level first undergo global demethylation and then remethylation and so on. So it's actually highly dynamic. How whether the methylation can still record lineage histories in such a dynamic modulation is also not clear. So we actually developed a method called methyltree to systematically solve all these problems. So we take the sparse single-cell DNA methylation data as input, and then we select the genomic regions, and in each region we calculate the average methylation level, and then we generate a similarity matrix between cells based on this methylation matrix, and then we do downstream lineage analysis to generate the lineage trees. So the crucial point here is that we only use the jointly observed CPG sites between two cells to calculate the similarity matrix in order to solve this sparsity problem. And to solve the second challenge, we recognizing that this is the similarity matrix for simulated data. And you can see that because of the cell differentiation, you have a lot of signatures. The first column is the clone identity, and then the second column is the cell type identity. You can see that in this simulated data, the cells are completely separated by the cell type identity. And we developed a method to separate the cell type signal and the lineage signal. After subtract the cell type signal, we can correctly reconstruct entire lineage histories in this simulated data. So then we applied it to a real experiment generated in our own lab. So here we took the stem cells from the mouse bone marrow, and then we add a LARI, which is the synthetic barcode, into the genome. And then we eventually expand and differentiate the cells for 6 days. And for each cell, we generate its LARI lineage barcode, DNA methylome, and transcriptome. So based on the single-cell transcriptome, we can do low-dimensional reduction and identify about 9 different cell types. They all have highly distinct gene expression. And however, using only the DNA methylation, we calculate the similarity matrix and found that this highly high similarity means that cells are similar in lineage, and indeed you can see that it corresponding to exactly the same genetic barcode from the LARI. And so actually, we have about 55 different clones, and this similarity matrix reconstructed all these 55 lineage barcodes accurately. So this is a tree that we generated using only the 21 multi-cell type clones, and it's also reconstruct all the clones 100%. So we down-sampled our data from original 13% genomic coverage to about 4 or 5% genomic coverage. You can see that the accuracy is still about 100%. This is, in each case, we use all possible genome-wide selections of the features to generate the result, and the result is actually quite robust to our selection of the genomic features. And another point is that if we use about 10,000 beans to generate the matrix, so you barely get any lineage reconstruction. accuracy. However, notice that it's actually very, very common in the field to use 100,000 bins to do this low dimension reduction with DMS data. So in our case, we actually focused on the much smaller bin, for example, 500 base pair, and this gives us almost 100% recovery. So we also test this algorithm in the human blood in vitro expansion data, and we see similarly that the reconstruction is actually 100%. And another interesting application to see the early embryo development, as we know that in early embryo development, cells undergo drastic demethylation. And however, you can see that in the full cell stage, so in this data, in this public data generated by Fu Chou's lab, so there are multiple different embryos, and each embryo they collected individual cells, and using our method, we can separate cells by the embryonic identity accurately. And also noting that there are only 4 cells here, so cells are separate into 2 groups. Each one has 2 cells exactly, corresponding to their division histories. And this is all human data, and you can see that also you can reconstruct the lineage history exactly, by separating cells by their donor identity. Also, we apply this to the cancer data from Fu Chou's lab, and you can see that we can 100% reconstruct the lineage based on the CMV identity. So, so this is a diagram showing the global methylation profile of a mammalian development system. So starting from the third zygote, you can see global demethylation and then remethylation and so on. And in germline, you have demethylation again. So we tested our method across a full spectrum of this process, starting from 2 cell, 4 cell, 8 cell, and so on, and also test the germline cells, somatic cells, and also homotic cells, and so on, and also other cell lines. So across all these spectrum, so our method reconstruct lineage history at about 100% accuracy. So, and also interesting to know that we observed that the temporal resolution could be close to individual divisions. And also, as I mentioned, by the way, so the cost of the generating single cell DMS data is actually continue to drop. So if we wanted to do both single cell DMS and gene expression, we can get about $8 per cell. And if we only just care about DMS, it can be even cheaper. So it's possible to generate thousands of cells in just maybe a few weeks. So we applied it to study the human embryo development. One interesting question there is that when the first cell fate decision happened to decide that a cell will become in the cell mice or surfactant, and applying it to a single embryo, and we generate a lineage tree, and based on lineage tree, we found that these cells, they come from the same 4-cell stage cell. It's the daughter cells from a 4-cell stage cell, and they all adopt this ICM, inner cell mice fate, suggesting that at the 4-cell stage, there's already a strong fate commitment happening there. So based on this and other data set, we conclude that there is this stochastic yet early commitment happening at the 4-cell stage. In another application, we are interested to see how many hematopoietic stem cell clones exist in the mouse bone marrow. So we applied it to our previous data from the Cell paper, and again we see this block-like structure corresponding to exactly the same clonal barcode introduced at the embryonic A10 stage. By counting the number of these blocks and combined with numerical estimation, we conclude that there are about 250 clones exist in the mouse bone marrow. And across 3 different replicates at different stages. And we also verified this using our Darling model, and it also see about like 300 different clones. I believe that this method opens the way to study human embryo development and brain and cancer and also other other systems as well. And lastly, I wanted to thank my collaborators for the Darling and the epigenetic memory work, and also my own team. Mongyang did the experiment, Ruijiang analyzed the data, and I developed the message tree method. So this paper has been accepted in Nature Methods. And also, I will present a poster in the poster section. Thank you very much.
Thank you, Shou-Wen. Questions from the audience?
Oh, great talk. I have a question. I'm curious about how you generate your DNA methylation data, the single-cell DNA methylation data. It's low input or high throughput?
So currently, we use single-cell bispecific, so it's the plate-based low-throughput method.
Oh.
Yeah.
Oh.
But we are thinking, we are trying a different method to try to scale it up.
So in Nature Methods, how you generate?
Using bispecific, yeah, single-cell bispecific.
Also low input.
Yeah.
Oh, thanks.
Hello.
Shem?
I think he was first. Yeah.
Thank you. Impressive talk. And I'm curious, what do you think, which intrinsic property of the DNA methylation that enable you to perform such lineage tracing? Is the statistic of the methylation and demethylation process of the liver-lipson signature or something else?
Yeah, so I mentioned earlier that the epimutation rate is around 10 to minus 3 per CpG site per division. So this epimutation it can be any reason. It's just the change of the methylation profile. And it is 10 to 6 orders of magnitude higher than the somatic mutation rate. So it generates abundance of information for do lineage tracing. So that is the underlying reason for using this for lineage tracing.
Okay, thank you.
Shou-Wen, I had a similar question. Just to clarify, if I give you a sample, any random sample, you can just do single-cell DNA methylation on it, use your algorithm, and then you can construct a tree. Is that right? You don't...
Yeah, so the key part here is that you don't need to do any labeling. You just need the primary samples. Yeah.
Right. So then I'm curious then, because cell types have similar cell types have similar DNA methylation. How does your algorithm zoom in on the lineage-based shared methylation and filter out the, okay, I guess the lineage and the cell type are broadly related, but still, at a very early stage, you could have the same cell type coming from different parts of that ontological tree, right?
So, as I mentioned, in principle, there is this confounder of the cell type signal. So in general,
so how do you handle that?
We basically, the trick is so one way to do that is maybe you can try to find this lineage specific CpG sites, right? Only focus on those and to reconstruct the tree. But we realized that it's actually a very non-trivial task. So instead, we first just use the whole genome DNA methylation to generate a similarity matrix. And then based on a phenotypic label, we can do computational regression to get rid of this cell type signal and then just leave the lineage information alone.
Okay, so you just regress it out.
Yeah.
Okay, got it. Thanks.
Thank you for your nice talk. So I remember you mentioned you also checked that on the human cells. So what kind of human blood cells? PBMC or BMMC? This is from the umbilical cord blood cells.
Okay.
And also we also checked all the human embryos and the germ cells and as well.
Okay, so my question, next question is about like how does this method could work for some leukemic cells differentiation, especially for the myeloid cell differentiation in like AML or CML?
Yeah, so as shown in this example, actually in tumor it's actually very easy because tumor have very strong, basically their DNA methylation profile changed too much. It's actually very easy task to do lineage tracing in tumor using DNA methylation profile. It's more challenging in normal cells because the change of the DNA methylation much more mild.
Okay, so how is the, how to say, the sensitivity? Like, can you also tell, for example, the difference between, let's say, the HSC in the leukemic cells may be more primitive or more quiescent?
Yeah, so I think the key issue here is the sampling, because if I could sample, if the cost can drop and then I can generate thousands or tens of thousands of cells for their DNA methylation, so that give me enough coverage of the lineage tree, and then I can answer questions like, for example, quiescence and proliferation and so on. So yeah, and also because in principle, I think in general, I think it's better to have joint modalities, both the DNA methylation and the gene expression, and then you can use the DNA methylation to record the lineage, right? And then you use gene expression to separate cell types, and that give you a single cell multi-arm lineage tracing, and use that you can, in principle, answer all the questions you wanted.
Thank you very much.
Yeah, beautiful talk. Can you give me some intuition why such a small bin size is actually giving you better resolution? Because I would say the smaller you get the bin, the more noise you would introduce, but it seems to be the opposite, right? So with 500 bases, you get really good accuracy. Why is that?
Yeah, great question. So if you think about epi mutation, so it's actually a stochastic mutation, right? If you average it over 100,000 bins, and then think about a central limit theorem, it basically reduces the signal to 0, right? So only when you look at very small bins, that give you, that you can retain the stochastic signal. And so that's why you have to go to the small bins. The question is now, if you have such small bins, then the sparsity is huge. How do you deal with that? So that's why I mentioned earlier here. So we only calculate the similarity matrix using the shared epi mutations between two cells. For example, if one cell has 5% genomic coverage, and then two cells can on average have only 0.225%, we only use that small fraction to calculate similarity.
Okay.
Yeah.
Thank you.
I'm curious how if you have genetic barcodes used to reconstruct lineages, then there's sort of a natural starting point of each clone, but the natural starting point of the methylation signal does not seem, defined at a particular time point that would necessarily match up with your genetic barcode. So why do the clones that you sort of reconstruct with the methylation signal have sort of the same natural timescale as the genetic barcode clones?
Yeah. Yeah, great question. So for example, you look at this. Here in this experiment, we took out individual cells from the bone marrow, right? So at this point, because the mice has like millions of cells, and then you took a small fraction of cells. So on average, each two cells have very different DNA methylation. Because of epi mutation, it generates a huge diversity inside the bone marrow. And then you randomly sampled about 500 of them, and then do in vitro study. And at least, on average, these 500 cells have very different DNA methylation. So that provides a good starting point. Let's separate the methylation profiles within the same clone, if that makes sense.
I'm still not sure. I'll ask you after.
Yeah.
Thanks.
Yeah, beautiful talk. It's really, really impressive. I'm very convinced that you can identify subclones. I think one of the things that is interesting with respect to lineage tracing is the idea of trying to find cell type of origin, right? Our neuroendocrine cells, the cell type of origin of small cell lung cancer and things like that. Can you explain how, is it possible to use this to get to cell type of origin? Because it seems like you have to try and find the normal cells that had generated your end product, which seems like a really hard thing to, or the branches of normal cells that eventually generated tumor, which sounds really hard to do. So are there ways to try and get cell type of origin using this kind of lineage tracing?
Yeah, it's a brilliant question. So I think in general, yes. one way to get around this is you can sample cells in the primary tumor and then get also samples from the metastatic sites, and that will help you to match whether this clone is starting from this region or not. And maybe a normal cell origin. So actually I'm optimistic because the DNA methylation has so much information there. So maybe there's a way to, bioinformatic way to identify some like special regions also and to help you to determine the origin, but we can talk offline. Yeah.
A very nice talk. I'm wondering the reason why only the DNA methylation is maintained during the long time clonal memory, but not gene expression or chromatin accessibility. Is it available to use hydromethylation to perform the lineage tracing or other DNA or RNA modifications like 6-methyl-A?
So the first question is why only the DNA methylation has this high memory, right?
Yeah.
So first of all, you can see that here the tracing time is 10 weeks. So there are already a lot of cell division going on. And to my understanding, so I think gene expression can also record memory, but in a much shorter time scale, maybe just in 1 or 2 cell cycle, or maybe a few more, but not too much longer, because it's a lot of like a transcriptional noise, and then this will destabilize the memory. And also, I think something similar is also for chromatin accessibility.
Okay.
And for your last question, so whether other modifications can also recall memory?
Yeah.
I believe so. So if as long as you have mutations, you have changes, and then this change, the frequency of this change is much lower, then that should work mathematically.
Okay, thank you.
Last question.
Hi, impressive. I wonder, once you regress out the cell type specific profile, I mean, I know that the nature of methylation is assumed to be kind of stochastic, but do you see enrichment in specific genomic regions of the remaining marks? And how do you test that in case? Is it truly stochastic or not?
So we did try to see where these IP mutations are enriched in the genomic region, and we found that it is enriched in the solo CPG sites, which is the CPG sites without any CPG neighboring. So because there's a correction mechanism, if there is a CPG island and then one of the CPG sites was mutated, and then this can be corrected by the information from the neighboring sites. But for WCG, solo CPG sites, I think if once you have IP mutation, it's very hard to correct that. To test whether this is truly stochastic, I mean, actually, I would argue that based on this DNA methylation data, right? So because the barcode is just stochastic random. You don't know like which cell has which barcode, right? So since that across all these data sets, we have seen that the DNA methylation profile match accurately to the random barcodes. It must be random. The IP mutation must be random.
Mm-hmm. Thanks.
Sorry, I have a quick question. As you mentioned that the method requires at least 5% genomic coverage and small beans. So my question is that the method is not fit for those high-throughput single-cell DNA methylation sequence method, right?
Sorry.
Those high-throughput DNA methylation sequence method with very low genomic coverage.
So you mean whether this method also works to high throughput single cell DNA methylation methods with low coverage?
Very genomic coverage.
If you can do targeted sequencing, yes, because all this method requires is that 2 cells share enough IP mutations, enough CPG size. For example, I found that if 2 cells have about 10,000 CPG size that are shared, then that should be sufficient. So if you can do enrichment to certain regions, then you don't have to go very high coverage.
Okay, thank you.
Thank you. Thank you, Shou-Wen. Brilliant work. So we will have a break for 15 minutes.